Dr. Etter is an assistant professor at the Department of Nutrition and Food Sciences, where she works on understanding the factors that lead to foodborne outbreaks from bacterial and human angles. She draws on her experience from homesteading in rural Wisconsin as a child, and is particularly interested in the biosecurity risks associated with homesteading and backyard farming, and in improving research and education to this under-reached group. Currently, Dr. Etter studies how common salmonella is in backyard laying hens and hatchling chicks, and what owners know about biosecurity and best practices to avoid catching salmonella from their birds. She works to determine the importance of sanitizer tolerance and biofilm formation capability to Listeria monocytogenes colonization of Vermont dairies and cheesemaking facilities, and understand whether bacterial stress is a key contributing factor to foodborne outbreaks of Salmonella and enterica. This talk will be about 30 minutes long with 15 minutes of Q&A at the end. I'll ask that you please hold your questions until the end. Welcome to our remote guests. If you're joining us via Zoom, you can enter your questions into the chat box at any time during the presentation. I'll turn over the floor to Andrea.
Thank you, Eurydice. So this work is actually largely done by Eurydice and a number of undergrad researchers in the lab, as well as a former master's student who now works at Diagnostics. But it has been a long process. I'm going to assume you guys aren't all microbiologists, but hopefully you can understand the main points without having a deep understanding of bacteria and bacterial genetics. So Listeria monocytogenes, if this name rings a bell, it might be because of a recent outbreak from boar's head deli meat, is a foodborne pathogen. It is rarely a causative agent of human illness. However, it is considered to be ubiquitous in the environment. If you look for Listeria, you will find it. And it has been found in lakes and streams and groundwater and produce and dairies and ruminants and on and on. We care about it because it tends to target the most vulnerable populations. A healthy individual may or may not get sick, and if they do get sick, it'll be very mild. It'll be like any other food poisoning. For someone who is under a year old or in the womb, in utero, someone over the age of 65, which the CDC considers old, pregnant, or immunocompromised by anything from diabetes to HIV/AIDS to organ transplants to cancer treatment. This can be a very, very serious infection because Listeria can cross from the intestine into the bloodstream, and from there it can get into the brain and cause meningitis and encephalitis, which are deadly often, as well as it can cross the placenta and infect the fetus in utero, leading to stillbirth or a newborn with really bad septicemia or brain infection. It is rare, so we estimate there are now only about 1,200 cases per year in the US, but it causes 172 deaths, which makes it the 4th most deadly pathogen in the US from food. Small but mighty, and if you get invasive listeriosis, there's an 85% chance you're going to be in the hospital, and about a 16% chance you'll die. One of the reasons it's such a thorn in the proverbial flesh for the food industry is that it's really hard, partly because it's ubiquitous, to keep it out of the food environment. It can be tracked in from the outside, it can be tracked in on raw product, it can come in through a variety of arenas, but once it's there, it has the tendency to stick around. And usually it does this through something called biofilm formation. If you've seen rocks in a stream covered with like a slime layer, that is a form of biofilm. There actually are not many biofilms that are quite that visible. If you had to unfortunately dig out your drain in your bathroom and there's a slime layer on the inside of your drain, that is also a biofilm. So is the pink ring that forms in the tub if you don't clean it regularly, and also plaque on our teeth, the biofilm. Listeria generally is going to form biofilms with other bacteria in the food processing environment in places that are hard to clean and or infrequently cleaned. It itself is considered a very poor biofilm former. The top image is Listeria growing on a stainless steel chip. The bottom image is Salmonella growing on a cantaloupe. You can see that one of these things is not like the other. The Salmonella is forming a much denser, thicker biofilm with a lot more 3D development. So there's actually a debate in the Listeria world about whether Listeria actually forms biofilms. I'm firmly on the side of yes, it can form a biofilm, but that is fairly strain dependent. So some Listeria will and some won't. Some will form really impressive for Listeria biofilms, and some of them will, look like the top picture. So because it is not a particularly good biofilm former, we do think it primarily is going to find friends to form a biofilm with. And there are a lot of them. Pseudomonas is a really common one in produce. But in the water system, Ralstonia is another option. And there are also a number of other ones that can form one. Listeria is also thought to be a really small percentage of most biofilms, so it's not like a major player, but it'll hang out in there, and as long as that biofilm is there, it is safe. I chose to look at cheese for a couple of reasons. One being that Listeria outbreaks have had a disproportionate number associated with cheese. And the other main thing is that artisan cheese is a major agricultural product in Vermont and very important to Vermont dairy sustainability and profitability. And we like our cheese, but Listeria also likes her cheese, so we need to figure out how to keep Listeria out of our cheese. All right, so I mentioned Ralstonia. Ralstonia is kind of the opposite of Listeria. It has a different makeup of its exterior, and it is opportunistic. It is generally not going to make you sick unless you're already very sick and your immune system just can't keep up. In that case, for people with cystic fibrosis or other serious infections, it can occasionally cause septicemia or bone infections or meningitis. But it's just emerging as a pathogen in hospitals. It is a very small pathogen. Ralstonia insidiosa, one of the species, is called insidiosa because it can get through the water filters. So you can have a 0.2 micron water filter, which is a really small filter that should filter everything out, and Ralstonia can get through that, which makes it really hard to keep out of your water systems. It also tends to show up in weird places like disinfectants and things with disinfectants. And like Listeria, it's got a very wide growth temperature. Listeria can grow in your refrigerator, it can grow in the human body. Ralstonia cannot quite grow in your refrigerator, but it can grow in, it can grow it from about like 50-ish, mid 50 degrees Fahrenheit to human body temperature. Both pathogens both bacteria are well adapted to low nutrient environments. Ralstonia particularly, there's not a lot of nutrients in a tap water system or water filtration system, and it will do fine. Listeria won't do quite as well, but it can cope. So Ralstonia, because it's so tiny and can fit through your filters, and it can do really well in low nutrient systems, and it really isn't picky about temperature, is pretty ubiquitous in potable water systems, including, unfortunately, the International Space Station, which is where one of our isolates comes from. And Ralstonia is interesting to us primarily because it is a really strong biofilm former. This is not it in a biofilm, but this is what it looks like, and it sort of naturally clumps together. There is research that has been published that Listeria will auto-aggregate in the presence of Ralstonia or even of supernatant from a broth that Ralstonia has grown in. So even if there's no Ralstonia present, if Ralstonia was there, something in that liquid will now help Listeria grow better. If you put Ralstonia and Listeria together on a surface, they will form a really dense biofilm with each other. Auto aggregation is usually on a water surface, so it's like rafts or little bundles, whereas a biofilm is traditionally considered to be attached to a solid surface. We don't understand how this works. There's been a bit of research. Nobody's quite figured it out. So there's a few options. Listeria could just add itself onto an existing biofilm of Ralstonia. It could integrate itself into the biofilm. Or Ralstonia could increase Listeria's EPS production. So EPS is like the sticky slime layer that you see on the outside of a biofilm in a drain or wherever, that is really important for the biofilm, both sticking around and being resistant to sanitizers such as bleach and Lysol and hydrogen peroxide and any number of other things we might use to try to get rid of it. There's also a possibility that this works by the bacteria talking to each other. So bacteria have this sort of chemical sonar system. And if you're closely enough related, you can hear the pings from the surrounding bacteria. And they use this to figure out how many other bacteria that they can communicate with are around. They actually won't usually try to cause an infection unless there's enough bacteria around. They won't form a biofilm. They kind of want to get a head count, a census, a quorum of how many other bacteria they can engage with are around before they do something that's going to be costly in terms of energy or expose them to like the immune system. So quorum sensing is very, very important for bacteria. And it's possible that Ralstonia can that Listeria can sense Ralstonia's quorum signaling, and that encourages it to do things it wouldn't do on its own. But we don't really know. All right, so I started this project, well, phase one of this project when I first started at UVM in 2019, looking at a set of Listeria monocytogenes that came from a Vermont artisan dairy that Kathy Donnelly, who was a professor here for a number of years, had collected from there. And this unfortunate dairy had this particular subtype of bacteria that they didn't see in any other plants. They tested a number of them, and it's actually a really rare subtype of Listeria. The few papers I could find on it, and there were like 4 of them, suggest that it's associated with ruminants such as cows. And it stuck around there for about over a 3-year period. So we had hypothesized that maybe it was an extra good biofilm former. And we did discover that overall it was unusually good at forming biofilms. And that particularly, if you stock it in nutrient poor conditions compared to optimal conditions, you would see fewer bacteria because they don't have enough energy to grow and divide. But you would see a fair amount of EPS or that slime layer, and you would see that the fact there were fewer cells didn't actually affect how robustly it could avoid being killed by sanitizers. So the top one is a nutrient poor biofilm of Listeria of this strain, and the bottom one is a nutrient rich. And you can see that there's definitely more bacteria on the bottom, but the top one has some like fuzzy bits, and that's the EPS. And so that's going to help keep the sanitizers from killing the bacteria. It also, something about attaching to surface also seems to make bacteria less able to be killed even if there isn't EPS. I don't know if it toughens their cell membrane or their cell structure or what it is, but they attach to a surface and they're almost instantly harder to kill. And we care about nutrient deficient surfaces from a food processing standpoint because these are areas we're generally not going to think about cleaning. So they might be a corner, might be the underside of equipment. It's something where we think, okay, well, nothing's going to grow there. There's no nutrients. It's not like the inside of a cheese vat. But bacteria are really clever and they will always exploit our laziness and our weakness. And so that's what they do. All right, so we knew that these weird ST191 Listeria were really good at forming biofilms by themselves, but bacteria do not like to hang out by themselves in the real world. So we wanted to see if maybe Ralstonia would be something that they would enjoy hanging out with, for lack of a better term. And Matt Wargo had a bunch of these Ralstonia, and he was interested in it. So we've been sort of coming at it from different angles. He's more interested in the clinical. We're interested in the food safety. He does a little more molecular. I do a little more applied, like what are the consequences? And it's a really nice partnership. The questions I'll be talking about today are, firstly, how do Listeria and Ralstonia interactions affect biofilm architecture? So the 3D structure, how much exopolymeric substance there is, and where the different species are. And is there a strain specificity? So if we look at multiple different isolates of these bacteria, like, do we see different results? Or is it always the same? And then does this actually lead to them being harder to get rid of because we can't use sanitizers effectively? So, brief overview. Eurydice is way more familiar with the gory details because she's been the one actually doing this. But we had 2 Ralstonia strains, one from the International Space Station potable water system where they had a biofilm, and one from a cystic fibrosis patient. Then we had 3 Listeria. These 2 were from our nasty rare strain. The other one was from a dairy, but it wasn't persistent. And we grow these either alone or in combinations on stainless steel chips that were the same grade of stainless steel that they use in food processing, with a nutrient poor condition, looking at both 11C, which is a cheese aging environment. It's kind of at the lower end of what a Ralstonia can do. That's where the temperature of cheese is aged at. And then at 22 degrees Celsius, which is roughly what a comfortable room temperature is. So like 68-ish in Fahrenheit. And we grow these up and we'd enumerate from the coupons and we use SEM. Specifically, what we do is we have these grown up 1, 3, and 10 days, and then we take the coupon and we replace the bacterial growth media every couple of days so they wouldn't run out of nutrients and get starved. And then we take them out of their coupons, rinse off any loose cells because the loose cells might behave differently, and then put them into liquid, either phosphate buffered saline, which is just a nice salt solution like what you might use in a hospital for IV, or a Lysol type sanitizer or sodium hypochlorite. And then we'd sonicate them and mix them with glass beads to remove the biofilm using sound waves and mechanical force. And then we take that and we dilute it out so we can actually count how many bacteria were there. In the first place from our control coupon and after sanitizer treatment. These coupons all started out in the same dish, so they had the same conditions, but then we split them up. And then Eurydice goes over to the microscopy core and spends hours and hours taking pictures of all of these. And she did a number of statistics. We look at the log counts because when we're talking about bacteria, we're usually talking about somewhere between 1 million and 1 billion bacteria per stainless steel coupon. So logarithms are a friend. And we look at how much that is reduced after sanitizers, and there's mixed effects and ANOVAs, et cetera. All right. So if we look at Listeria growing in an environment where there aren't a lot of nutrients, we do see that it is going to grow better in the presence of Ralstonia than it is on its own, based on like how many bacteria are there. This is particularly true if you grow it at 11C, which is actually a pretty comfortable temperature for Listeria. This is all the different strains mixed together. You can see it starts off pretty low. So at 24 hours, we have between 10 to the 5th and 10 to the 6th bacteria per coupon. By day 10, there's a big difference between the single species coupon in gray, where we have 6.5 log, and the dual species coupon in black, where we have more than 7. So there's about 50 times more bacteria if you put in Ralstonia, which is interesting because you think Ralstonia and Listeria would be competing for nutrients, not feeding each other. If we look at room temperature, we see a big difference until we hit day 10, essentially, and then it's actually there's no difference. But partially this is because bacteria in general are going to be happier growing at room temperature than at 10 degrees Celsius. Everything moves faster, enzymes, bacteria, etc. And then before I show you the dual species, it's helpful to understand where the bacteria start from here. So this is strain 1 of Listeria at 11 degrees Celsius. The whole coupon doesn't look like this. We have spots where it's really dense, and it's forming these nice 3D clumps. And you can see there's some long bacteria and some short bacteria, and there's sections that have nothing. If we look at 22 degrees Celsius, we see kind of the same thing. Here's a big clump of bacteria. They're covered in EPS. You can see some not covered. And then there's areas where there's nothing. Strain 10 is one of our persistent strains, and this looks like it's flat, but this is actually bacteria covered in that slime layer. And if we zoom in a little, we can see more of that slime here. These long strands of it sticking in on there. At 22 degrees Celsius, we don't see as much of that. So this strain seems to maybe do a little bit better. If we look at 19, which is technically very genetically similar to 10, there's a lot more little clumps. It's just not as good of a biofilm former in general. There's probably minor genetic differences between 10 and 19 that could explain it. Need to look into that. And we do see slightly bigger clumps if we put it at room temperature. But while this strain is particularly impressive, these two strains are particularly impressive for Listeria at 22. Only this strain, number 9, does particularly well at 22 for Listeria. Ralstonia, if we grow it by itself on stainless steel, you can see that this strain is possibly a little stressed. These super long rods mean the bacteria it's growing really long and not dividing, and they usually do that when they're unhappy. But this strain, this one is from the cystic fibrosis patient, and that's one's from the International Space Station. This one is doing pretty well. It's got a lot of that wet cobwebby slime and a number of clumps. If we look at 22 degrees Celsius, our water system strain from NASA, this is more what it should look like. It's pretty dense. And this one, the cystic fibrosis one, is fine, but it's not as exciting, shall we say. And one thing I did find out about the NASA strain is that it basically had like no more than 5 degrees temperature variation for like a 10-year period. So it was really well adapted to more of a room temperature scenario. And the cystic fibrosis strain, probably not as much. All right. So just refreshing our memory, this is Listeria by itself. If we put Listeria with Ralstonia, top row is 11 degrees Celsius. And these are different combinations. We didn't have good pictures for all of the combinations. You can see at 11 degrees Celsius, number 9 and the space station strain, they have a lot of EPS, but maybe not a ton of bacteria by themselves. If we look at this at 22 degrees Celsius, this is clearly a lot happier bacteria. Though 9 works well with both types of Ralstonia, and that's a pretty dense bit of biofilm right there. You can see the cracks in the individual bacteria. And that's after 24 hours of growth. By day 3 at 11C, this is thicker. This is a different strain. And the main one, this is our cystic fibrosis patient, and this is LM10, which is a different strain than LM9. But we can see that the water system strain does okay by day 3. It's sort of adapting. And this is pretty great for Listeria. LM10 and the water system strain at room temperature, this is already getting to the point where it's starting to disintegrate a little bit. You can see the edges peeling off. That's the final stage of a biofilm. So it's really big, it's really mature. It's going to be really hard to kill at that stage. And it's overall a happier biofilm further along than at 11C. And if we look at day 10, this strain, this biofilm is still pretty robust. You can see bits where it is peeling. These are all Listeria and Ralstonia strains sort of curling up on the edges. And you can see there's a bit of peeling, but at 11C, it's a lot more, there's a lot more integrity. Whereas if you look at 22 degrees Celsius, there's big patches that are peeling off. So this is a strain that is this is a biofilm that is disintegrating, and it's going to go and reseed a new biofilm elsewhere if the cycle were to continue. So it really, the temperature is pushing the development of the cycle along faster at 22. And then I have a accelerated master's student who used the fact that Listeria and Ralstonia have a different cell structure to use a dye-based method. So this is a gram stain, and basically in the gram stain, some bacteria will end up pink and some will end up purple, depending on how their outer walls are constructed. Listeria ends up purple, Ralstonia ends up pink. As you can see, there's slight differences in how the Ralstonia arrange themselves. This is very, very fluffy, this 891 from a cystic fibrosis, and this one is a little bit more stringy. But overall, we tend to see Listeria on top of the Ralstonia in general. And Listeria on its own tends to form these big cobwebby things with lots of big holes. Chains and honeycombs is what they're described as. And that's at 11C. If we look at 22 degrees Celsius, so room temperature, we see sort of the same thing. The water system strain is a little bit fluffier than it was at 11, but it's still pretty fluffy. And then if we look at these together, it looks like Listeria is still possibly on top of Ralstonia, but the arrangement is closer to the Listeria and to the Ralstonia. So maybe Ralstonia is following Listeria's lead a little bit more. I actually had my student try to give Listeria a head start and see if it changed things up. He says he doesn't think it does, but he didn't have that analysis quite finished. So we gave Listeria an 8-hour head start, and it seemed like Ralstonia was still kind of changing the way Listeria attached to things. Very strange. So one of the things we were interested in is maybe Ralstonia's presence encourages Listeria to make more exopolymeric substance of the slime layer. And maybe this is going to make the EPS look more like Listeria. And so we use this dye called ruthenium red that attaches to carbohydrates in the exopolymeric substance. And we figured, and you can use a spectrophotometer to measure the amount of dye that is present, or basically the amount of dye that's been pulled out of it. And we kind of thought we'd see that when you put Listeria and Ralstonia together, you get more EPS, more of this ruthenium red OD than you would with either by themselves. Unfortunately, we kind of saw the opposite. So if you put Ralstonia by itself, it makes a little bit of this carbohydrate. If you put Listeria by itself, it makes a lot of this carbohydrate. And if you put them together, we get less than we do with either. And so that was not really what we were expecting. And we're not really sure why. but there are lots of carbohydrates and not all of them bind this dye. So maybe it's just making a different type of carbohydrate that doesn't bind this dye. The one where it does change is, interestingly enough, this 19 and 891 at 11 degrees. We see that it will follow, it actually makes more EPS than either of them on their own. So that one does its own thing. Again, we don't know why. All right, if we look at 22 degrees Celsius, we see that the bacteria basically stop making a lot of EPS after 1 day. So it's early on in the biofilm process. And once again, like if we put them together, it tanks. And interestingly enough, at 22 degrees Celsius, the difference in this carbohydrate is very negligible between Listeria and Ralstonia. We don't know why, again. So answering question 1, we see that Listeria sits on top of Ralstonia, but we need a different method to figure out what if anything is changing in the EPS. We can see a lot of EPS on scanning electron microscopy, but unfortunately Listeria and Ralstonia look identical on SEM, so you can't tell them apart. So we're gonna look at some more dyes and see if there's anything different and use fluorescence microscopy next. All right, so we were really interested in how this would play out in the real world. So if we look at quaternary ammonia compounds like Lysol, and then sodium hypochlorite, which is bleach, and just Listeria growing on its own at 22 degrees Celsius, we can see there's a lot of range and effectiveness. So this is log reduction, which is how many bacteria do we lose when we treat it with a sanitizer? Ideally, for EPA effectiveness data, we should see 6 log, or like 1 million fold reduction, like 1 million times reduction, 10 to the 6th. We are not even seeing 5 log. For this strain, we get close, but the error bars are enormous. Unfortunately, biofilm formation is a very phenotypic process, which means it does different things on different days. So we have to repeat our experiments over and over and over again because the bacteria do not do the same thing on multiple different days for whatever reason. And we also know that Lysol isn't terribly effective on biofilms, but there is a wide range and variation. For sodium hypochlorite, we overall see it as more effective. It is an oxidizing agent, so it will react with anything in your biofilm. And you just have to use enough bleach that it will still be active by the time it reaches the bacteria underneath the EPS. But if we add the bacteria together in combinations, so our Listeria 9 and Ralstonia 602, Listeria 9 and Ralstonia 891, Listeria 10 and Ralstonia 602, Listeria 10 and Ralstonia 891. We do see that the quack reductions, the average is going down. So it's working overall less well, but the error bars are still pretty big. And if we look at bleach, we see that there's a definite strain effect, but also particularly for this strain, if we combine it with Ralstonia, bleach basically stops being effective. So we're removing 10% of our bacteria now, or 90% of our bacteria, but you want to have a 99.9999% efficacy at least, and we're not hitting anywhere near that. So if you calculate it out, basically we go from having 1.5 to 3.5 log reduction in single to having 0.5 to 4 in dual. And for sodium hypochlorite, the reduction is a lot worse. I think that's because of the EPS, because bleach doesn't care what it attacks. It just goes berserk and attacks everything. And if there's a lot of EPS, it'll run out of efficacy before it hits the bacteria. And then there's lots of bacteria left alive. And we do see a lot of strain variants. I'm gonna skip through the slide a little bit because I think we're running out of time. But the strains respond to sanitizers differently depending on, and also so to respond to sanitizers differently depending on which Ralstonia they're paired with. So there is a lot of strain variation effect here. All right, and if we want to visualize this, this is a Listeria biofilm 10 days, Listeria Ralstonia biofilm 10 days old, grown at 22 degrees Celsius. And you know, it's definitely peeling, but it's still pretty robust. If we treat this with sodium hypochlorite, this is after treatment with sodium hypochlorite. And my one sentence summary of this would be nothing happened. These bacteria can probably regrow really well. And I actually have an undergrad who's doing a DUR honors thesis to find out how quickly these regrow. So not great if you're trying to get rid of bacteria. All right, so summarizing the research questions, we do see that combining Listeria with Ralstonia means that Listeria grows better in terms of cell number. The EPS changes are unclear, and Listeria usually sits on top with Ralstonia, mostly driving the arrangement and of the structure of the biofilm. Could that change? Yes. Is there strain specificity? Absolutely. Overall, it seems like the cystic fibrosis patient sample isolate is better adapted to colder temperatures than the space station one, but that's probably because the space station one never had to adapt, didn't have as many stressors. But we also see that even if we look at two very closely related Listeria strains, there are some differences in how they respond, both growing by themselves and paired with Ralstonia. Yeah, so I'll probably do a single nucleotide polymorphism comparison when I have a few minutes one of these years. And then does this actually make an effect in the real world? Yes. But there is a substantial strain variance. So really we don't want Listeria combining with Ralstonia. But it also depends which Listeria and which Ralstonia we have involved. The big takeaways is that you really want to get rid of your biofilms early before it's too late. You can get rid of a really dense biofilm by extensive scrubbing and enzymatic treatment and repeated sanitizer application, but you will probably damage your surface, and Listeria loves the little micro abrasions on the surface. It makes great hidey holes for it. And then you just build this nice dense biofilm right where you had the previous one. I sometimes joke that if you realize you have a really bad biofilm problem in your food manufacturing environment, you should probably just nuke the place from orbit, because you'll just never get rid of it. Maybe with extensive renovations followed, like tearing out whatever has the biofilm, followed by extensive cleaning, followed by extensive testing, but it's going to be a pain, a massive pain. We still don't know what the mechanism is. I'm applying for a USDA grant on this cycle and hoping to do some more work on this. There's been a lot of attempts to answer this, but most of them have been closer to human body temperature, which is not really relevant for anything outside the human body. And yes, and yet most of these are being published in food journals. I do not understand why. All right, the next thing is, if we can figure out the mechanism, is there a way to disrupt this? This is the gold standard. Is there, if it's quorum sensing, can we put in an anti-quorum sensing molecule? If it's a nutrient, is there something we can apply that will stop Ralstonia from making the nutrient? And we really are curious what other members, bacterial members, we should be adding to this that would be relevant to drains and water systems, and what their effect would be on this. But anytime you add more bacteria, it gets exponentially more complicated. And finally, one question that my collaborator, Matt Wargo, is particularly interested in is, what does this combination do to Ralstonia antibiotic resistance? Listeria doesn't do antibiotic resistance very well. So it's probably going to be more resistant if you put it with Ralstonia, but in general, you can treat Listeria infections with penicillins, and you can't do that for Ralstonia. And we also know that Ralstonia is extremely sanitizer tolerant, so this is probably not making things substantially worse, and it's not a foodborne pathogen, so from the food safety angle, we don't care. All right, so I have had a lot of people work on this particular dual species part of the project. There have been 6 undergrads, and there's another 3 new ones who just joined. And then I was on Sophia Unger's committee when she did her distinguished undergrad research thesis. And that's been great. And I wouldn't have these strains if Kathy Donnelly hadn't had good enough relationships with the artisan cheese community that they were willing to trust her to find Listeria in their systems. So with that, I will take any questions. Hopefully you guys are able to understand some of this without having a huge micro background. Yeah.
Is there, so you still don't know if it's the structure.
Mm-hmm.
Is there any way to simulate the same kind of structure mechanically, like etching on a piece of metal or something, so that you could kind of see if there's a huge difference? They're just using Ralstonia as like a scaffolding.
That's an interesting idea. So we know Listeria on its own will find like the little scratches and nooks in the stainless steel and attach to that the rough spots. Most bacteria do that. I don't think you get the 3D structure that way. And that's a major thing. I don't think it affects cell numbers. So I do think there's like a biological, biochemical thing going on. But certainly the surface, one of the things I don't like about bouncing back and forth between glass slides and stainless steel is it is known that surface hydrophobicity affects biofilms, and like it's different between stainless steel and glass. Mm-hmm. But we can't visualize Listeria and Ralstonia effectively on stainless steel, if we don't have it on a glass background, because they are shaped the exact same and about the same length and all of that. As far as I know, there's no way to do light microscopy or fluorescence microscopy on stainless steel.
So maybe you could do something interesting with etching in glass.
Glass, yeah, maybe an etched glass surface would be more similar.
Or at least try different, if you can.
Yeah, different types of glass slides.
It's fascinating. I found it fascinating that they can communicate. Perhaps with each other, right?
There is some evidence that they communicate, but it's probably also condition specific, and most of the studies have been done at much higher temperature.
Well, it's always fascinated me, like why some years in Lake Champlain, the blooms are toxic and other years they're not. Do you think it's the same? You said that if they get to a stressed situation, you think they turn something on that makes the environment toxic. What's the evolutionary advantage to doing that? Is that you're killing off your competitors?
So cyanobacteria is going to be pretty different.
No, I mean, I'm not doing that. I just thought it was really fascinating.
Yeah,
you said that you observed this.
Right. Well, I mean, that's known broadly speaking. I would imagine that some of it is probably form sensing in Lake Champlain.
No, but like in this system, what might be the advantage? They're getting crowded, right? And so they do something that's toxic to what? Like not
to the human body usually. I mean, they can produce toxins against each other as well. Not sure if Listeria does that as much. So like some of the bacteria in fermented foods like yogurt and cheese will make things that will kill off Listeria. I'm not sure about Listeria's ability to do that.
And when they're doing this, they're in the biofilm. So is there an advantage to producing a toxin that
you kill off your competitors
and leaves more room?
Yeah. And then you can eat your competitors for parts. There's also that. I mean, in the human body, it's unclear if the Listeria is in a biofilm or not necessarily, but it's not going to start invading cells as much until there's enough bacteria that it's likely to be successful. They'll sort of just hang out and try to keep a low profile.
But when you say, I'm sorry, other
Go
people
for it.
should ask questions.
Yeah.
So what did Boar's Head do after the outbreak?
I heard a very interesting talk at the International Association for Food Protection annual meeting by someone who's on the food safety board of Boar's Head, who insisted that it was not Boar's Head's fault at all. And it was, obviously, it was bad luck, and it was a tiny area. They did all the right things. I think they initiated a very extensive cleaning and sanitation campaign. I can't remember if they replaced any equipment. It was liverwurst that was contaminated. But they would have had to do some extensive follow-up testing, and obviously they've been doing a ton of ads trying to restore public confidence. So, you know, hopefully they change something more substantial than just running a bunch of ads.
From your scientific standpoint, would you like purchase Boar's Head in that specific like meat afterwards, or would you just be like, no, because
So
they
it-
probably didn't clean it
It
correctly or something?
depends. Sometimes if a company has gone under the microscope badly enough, they'll be like super, super careful.
Right.
And then they're actually safer because they're taking it super seriously. I'm pregnant, so I'm not eating deli meat at all, you know, unless it's heated for some reason. I definitely wouldn't buy Boar's Head right now. The fact that they were trying to insist they did nothing wrong and rings a few alarm bells. Because generally it suggests to me that there is a decent possibility it could happen again, that they may not have taken it quite seriously enough. I'm sure that people have kept an eye on it, but like there was a company that had an outbreak associated with ice cream that went on for years and years, and they were able to successfully reopen. So they had to do extensive renovations and extensive testing. And I think they had to get the FDA's permission to reopen. I do teach an outbreak investigation course every other spring for foodborne outbreaks, and yeah, everybody had known there was a risk of a ice cream associated outbreak, but nobody was willing to invest in the testing and prevention, short of like there being a compelling financial reason. Yeah, and that was probably, there were leaking pipes and things, and there was probably a biofilm involved somewhere. It's like they tested 2,000 single scoop ice cream things going to hospitals and found Listeria in like all but a couple in very low levels, which is not a normal scenario, shall we say. Other questions?
I'm assuming there's no other easy way other than scrubbing or burning your place down. Heat or cold or UV or.
So Listeria is very sensitive to heat, so it's really only a food safety hazard for things that are not going to be typically reheated before eating.
But could you like heat your, I'm just thinking if you
You
had to clean the
could,
place, you know,
but,
could you-
Saw it.
But if you heated it, you potentially be like caramelizing the sugars on, and then they're going to form a rough surface that's easier for bacteria to attach to. So really, you know, the first step is like enzymatic treatment and manual scrubbing, followed by sanitation and lots of testing. And then if you keep having issues, then you probably need to replace whatever is popping positive all the time. But of course, nobody, you know, usually wants to do that. So they'll try to come up with a way to avoid the capital investment.
Fun talk.
Well, thank you.
You're welcome. Hey.